Hello, welcome to the genetics unit lecture video number 1. Today's lecture will be focusing on terminology and vocabulary that you need to be successful in this chapter. Let's begin with some terms. Heredity. Well, this unit is genetics, and genetics is the study of heredity. So what is heredity? Well, we need to know how information is passed on from parent to their offspring. So how do mom and dad, for example, how'd they make you? And when they made you, why do you have resemblances to your parents? That's what heredity is all about. And what does it come down to? It comes down to the chromosomes that your parents gave you. Mom gave you half your chromosomes, dad gave you the other half. So you have a total 46, 23 pairs. Why do we call them pairs? Well, there's chromosome number 1. You got 2 of those. One of them came from mom, one of them came from dad. Chromosome number 2, one of them came from mom, one of them came from dad. When you get to chromosome pair number 23, those are your sex chromosomes. You either got an X from your parents, you got an XX or an XY. XX would make you a female, XY would make you male. Okay, so chromosomes are passed on from parents to their children, and they carry the hereditary or genetic information. How do they pass it down? They have sections of these chromosomes. So this is one chromosome. They kind of look like an X. They're actually 2 pieces connected to each other by this point called a centromere. So it's 2 pieces technically connected to each other, and they have genes on them. And genes are sections that code for a characteristic, or they code for a protein, or they make a certain trait. This is why you have blue eye color, or brown eye color. You have certain genes that have made you that way. Humans have about 30,000 genes approximately. That's about how many genes a human has. And you have 46 total chromosomes. Okay, 23 came from mom, 23 came from dad. This gentleman named Gregor Mendel, he was an Austrian monk. Why do we tell you about him? Well, because he is known as the father of genetics. His ideas have passed the test of time. Your genetics crosses, you're going to be doing genetics crosses, these things called Punnett squares. Some of you have done them before. And he is the basis of all that information, where it all comes from. What did he determine? Well, he figured out some major things. He figured out that you receive 2 factors. The reason he calls them factors is they did not know about genes in his time. So, you're talking like an early time period where we haven't even discovered DNA. We don't know what chromosomes are. We don't know what DNA is yet. But he figured out that 2 factors, 2 things controlled a given trait. What do we know now? We know that 2 genes, and another word for a gene is an allele. We know that 2 genes now can act and give you a specific trait. One of those factors, one of those genes came from mom, one of them came from dad, and we just talked about that a second ago. All right. How did he figure this out? Well, what he did is he took pea plants. So Mendel's known for having pea plants, and he crossed them with each other. He mated them with each other, and then he kept looking at their offspring and counting and using math and statistics to determine what was going on and try to give a guess or a probability of what the likelihood of their offspring would look like. So what did he look at? He looked at things like the seed shape, the seed color, flower color, pod shape, pod color, okay? All kinds of things. Flower position, stem height, you name it. He looked at all kinds of things. And then what did he discover? He discovered that some of them, those characteristics would cover up other characteristics. The ones that did the covering up, he called them dominant. The ones that could cover were dominant. The ones that couldn't cover up another characteristic, he called them recessive. So we learned that he came up with some fantastic ideas. 2 genes, one from each parent. He figured out by counting, you make these Punnett squares, and you can figure out probability based off of the likelihood something should happen. He came up with the idea of dominant, something covering up another thing being recessive. So his ideas have passed the test of time. He is the father of genetics. An allele. Okay, so this is another word that is used in genetics, and this word means the gene. There's different versions of gene. For example, there's an eye color gene. And that eye color gene, you could get a brown eye color gene, or you could get a blue or gray or green. There's different kinds of eye color genes. So what this is telling you is that the different versions of the eye color gene are called alleles. So they're basically slightly different genes. All right, that's what alleles are. Dominant genes, things that can cover up or hide or mask something else. In humans, here's some common ones. Having a cleft chin dominates no cleft chin. So if you have a cleft chin, then you can't see it. I have a beard. I don't have a cleft chin, so I am recessive for cleft chin. A widow's peak is when your hair comes to a point. That is dominant over no widow's peak. Dimples are dominant over no dimples. Brown or black, dark hair dominates that light blonde hair. Freckles dominate no freckles. Brown eyes dominates lighter colored eyes, blue eyes, for example. Free earlobes, check your ears. Are they attached, straight attached, or do they have a hanging earlobe? Free earlobes dominate recessive earlobes. So there's shows you examples in humans. So the recessive gene, this thing. So if someone gave their child one free earlobe gene, one attached earlobe gene, well, that dominant gene would cover up that recessive gene. So their child would be born with free earlobes. Okay, so that's how this kind of works. Zygote, a fertilized egg. So this is when you take an egg and you fertilize it. So you take sperm and egg. Sperm have 23 chromosomes. Eggs have 23 chromosomes. When you bring them together, the zygote has 46 chromosomes. Okay, so for a human, that is a human zygote, 46 chromosomes, a human sperm. So dad can give their kid 23, half of their chromosomes. Mom gives 23, half of her chromosomes. Mom and dad combine and make a zygote, 46 chromosomes. So you cannot pass down, I already mentioned this earlier, you cannot pass down all of your DNA. You're only going to pass down half of your DNA. So when you do this thing called meiosis, and what is meiosis? Meiosis is a process that makes sperm and egg, sex cells for humans or any other organism. Meiosis, what does it do to the number of chromosomes that you can pass down? It divides them in half. So for example, the sex cells of a male called sperm have 23 chromosomes. Normal cells have 46. So they started out with 46, did meiosis and made cells with 23. So you can only give your kid half of your DNA. That's called the law of segregation. If you got a pair of genes, for example, let's say you get a capital A on one chromosome and you got a lowercase a on another chromosome, you can't give your kid both of those. You're going to give your kid one of them. The law of segregation is in effect. So you can give your kid possibly just the capital A or maybe just the lowercase a. This would be a dominant gene. This is a recessive gene. The lowercase letter shows it to you. What are gametes? Sex cells. So this is showing you can only give your kids half your DNA in your sex cells. And that's the law of segregation. The law of independent assortment. Now, this is kind of a complicated one. I explain it more when we do the actual genetics problems, because this doesn't make a whole lot of sense to you by looking at it right now. But you're going to be doing genetics problems for several days, 5 days in a row of genetics problems. And on the third day, you're going to see a large genetics problem, this big old box. And that is basically this big old box. What are these letters? Well, these letters are half of an organism's DNA. So for example, let's say an organism this is the DNA of the organism, okay? Mom and dad came together, their sex cells came together, the A's came together, and the B's came together, and they made an organism that has capital A, lowercase a dominant and a recessive, capital B, lowercase b, a dominant and a recessive. Well, this person here, when they make babies, they can only give their kids half their DNA. That's law of segregation, okay? But they can't, the way they pass their DNA, it's a random, it's an independently, these A's, they sort themselves independently of those B's. Well, what does that mean? Let me give you an example. Capital A or uppercase A could combine with uppercase B. There it is right there, okay? Uppercase A could combine with lowercase b. There it is there. That's two ways the uppercase A could sort itself. Now the lowercase a could sort itself with the uppercase b. That's right there. The lowercase a can sort itself with the lowercase b, and that is right there. So what it means is you need to give all the possible ways these A genes can sort themselves with these B genes. So it is called the law of independent assortment. So it involves a law of segregation because you separate them in half. These are half. Start with 4, you end with 2. All these have 2. Okay. All of these have 2. You start with 4, you end with 2. Okay. So that's law of segregation. But the law of independent assortment shows you the ways these different genes could sort themselves with the other genes. All right. So it's a little complicated, but it'll make more sense when we do some genetics crosses. Terms. These are important terms when you do genetics crosses. You need to know what homozygous, heterozygous carriers mean. You just have to know what they mean. Otherwise, genetics problems will be very difficult for you when we begin them on the next unit, in the next day's lesson. Homozygous means same, exactly the same. If they're homozygous dominant, they're both capital. If they're homozygous recessive, they're both lowercase. Real simple. Nothing complicated here. You just need to know that homozygous means same. Not just the same letter. It has to be exactly the same, as in all capital, all lowercase. Okay? So I'm not just saying the same letter. No. The letter has to be uppercase completely or lowercase completely to be homozygous. It has to be exactly the same. All right? Heterozygous means 2 different genes. Another word for genes is an allele. I use it on purpose to show you that that word can be used as a gene or it can be used allele. It means the same thing. So 2 different alleles. Usually, but not always, one dominant and one recessive. So for example, capital A, uppercase A, lowercase a. They're different. They're not exactly the same. Heterozygous. Uppercase H, lowercase h. They are different. Heterozygous. Now, you're going to learn these later on. It'll be I believe these are our 4th genetics cross, blood type. See this right here? This is an I with a superscript capital A and then a lowercase I. These are different. This is heterozygous. Okay. This one here, this is B type of blood. One way to be B type of blood. Okay. This is AB blood. Heterozygous because they're not exactly the same. Oh, males are XY, females are XX. XX is homozygous. XY is heterozygous. They're different. Sometimes you'll see a letter like an H with a superscript that is different, a W and a T. They're different. Heterozygous. Okay, so it's not always the same letter. It could be different letters also. Carriers. Now, what is a carrier? Now, a carrier, by definition, students get they make it more complicated than it is, and they get confused with it. A carrier is someone that does not show a characteristic. So let me give you an example. There's diseases out there that you could have that you don't have. You do not have the disease. You're healthy, but you have a recessive gene that you have in your DNA, and you can pass it on to your child. And if the person you marry or mate with and you produce babies with also has that recessive gene hidden in their DNA, and you both pass it on, then your child could be born with a disease, and you and your spouse or you and your partner may have been disease free. So you did not show the disease, but you pass it on to your babies. That would call you a carrier. Here's another way. Let's keep it simple. You could have brown eyes and your spouse could have brown eyes, but you guys could have a blue-eye gene covered up, a recessive blue-eye gene covered up by your brown gene. And then you and your spouse could mate and produce a baby with blue eyes. So it is possible to have a blue-eyed child and to have brown-eyed parents. You do not have blue eyes, but you carried the blue-eyed gene that you passed on to your offspring. So you did not have blue eyes. A carrier does not have it, but they can pass it on to their offspring. We're going to learn when we do genetics crosses that there's a certain type of genetics cross called a sex-linked trait, and only females can carry them. Okay, males cannot carry those traits. Males can and females can be carriers of things that are not sex-linked. If it's not sex-linked, males and females can both carry it. Genotype. You need to know what this means. So many of you will fall prey to trick questions on the test. When I ask you a genotype question or a phenotype question, you're going to not remember exactly what it means, and you're going to guess and mix them up. Genotype, this is the actual letters that an organism has. So capital A, genotype. Capital A, lowercase a, heterozygous, genotype. Lowercase a, homozygous recessive, that is called genotype. Okay, so if you know the letters, the actual letters, the genes, genotype. Phenotype is what the characteristic is. So if I say they're a female, okay, female, even though we know females are XX, look, there's an XX. This is a female picture of a female's chromosome. This is called a karyotype right here. Even though we know a female has an X and an X, by saying female, we are not telling you their genes. We are telling you the phenotype, the characteristic, what the trait is, the trait that is being expressed. If I say to you, you have brown hair, I gave you a phenotype. If I say to you, you have blue eyes, I gave you a characteristic. I gave you a phenotype. Okay. If I say to you, you have a capital B and a lowercase b, genotype. Okay. That's the difference. Exact letters, genotype, characteristics, traits, how they express themselves. That term express, that's the characteristic you're seeing, what the organism looks like. Autosomes. So this is a picture of chromosomes. There is, if you count them, you'll count 46. And if you notice, there is no Y. There's 2 X's. That makes this a female karyotype. All right. Now, the last set here are the sex chromosomes. All right. Are the sex chromosomes. This is the very last set. All the other ones are not sex chromosomes, so they are called autosomes. All right. Now, you may notice there's an issue with this karyotype. This person here has 1, 2, 3 instead of a pair. So now if you actually counted, I'm sorry, I might have said 46, or if you actually counted, this person has 47 chromosomes actually. So this is a diseased individual, and they have a disease I'm going to talk about in a little bit. So all the ones before the sex chromosomes are called autosomes. Okay. If they're a healthy individual, they should have 44. This person, 45, not a healthy individual. Okay. The last pairs, if they're a healthy individual, they are called chromosome 45 and 46 for a healthy person. And those would be this person here, 2 X's, is a female. Now, we discovered many years ago that we have, and I mentioned earlier, there's about 30,000 genes a human possesses. And how did we find that out? Well, we created chromosome maps of our DNA. So biotech firms took all the chromosomes that humans have and they mapped them. So what does that mean? They figured out where the specific genes that control certain things are located on the different chromosomes. So you have 23 different kinds of chromosomes, and each chromosome has a pair. So you have 2 of each kind. So there's 23 kinds of chromosomes, and you have 2 of each kind. That makes 46 chromosomes. So scientists have mapped and figured out how far apart, where they're located, and what they do. Okay, so these are chromosome maps. What do chromosomes maps tell us? They tell us things about genes that could be linked. Well, what does that mean? Well, if you look at this chromosome map, you'll notice that gene 1 is over here, gene 2 is way down here, gene 3 is close to gene 2, and gene 4 is down here. Okay, some genes are linked. That means they're inherited together. Let me give you a common one, and it'll make more sense to you in real life. You may notice people who have red hair commonly have freckles and lighter skin. It's like they all go together. So you'll notice someone who has red hair, you'll notice, whoa, they have freckles. You almost always, not necessarily always, but almost always, red hair, freckles, and fair complected skin. Not always, though. Okay. There is people who have red hair who have darker skin. I understand that. And there is people with red hair who don't have freckles, but commonly you'll see people red hair, freckles, light, fair colored skin. Well, that's because all those genes are really close to each other that control those characteristics. And people generally inherit them together. That means they're linked. Well, why? Why are they inherited together? Well, there's this process of dividing your chromosomes in half. You cannot give your baby all of your DNA. You can only give your baby half of your DNA. Okay. So pretend this is dad's chromosome you receive. Let's just say this is chromosome number 1, the first chromosome. Then this is your pair. So this is chromosome number 1 from dad. This is chromosome number 1 from 2. So let me go back a picture. See how chromosome number 1, there's 2 of them. Chromosome number 2, there's 2 of them. Chromosome number 3, 4. Down here we have 3 of them. That's a problem. But you're supposed to have 2 of them for each number. Okay? So let's pretend this is chromosome number 1. This blue one could represent the one you got from dad. This red one will represent the one you got from your mom. So your mom's chromosome, your dad's chromosome, they make a pair called a homologous pair of chromosomes. So you have a homologous pair, that is chromosome number 1. They're both chromosome number 1, one from mom, one from dad. Well, there's this thing that takes place called meiosis. You cannot give your kid all of your DNA. Remember, law of segregation, you can only give them half of your DNA. So meiosis takes place, and you're going to only pass down one of these chromosomes. Well, before you give your chromosome to your sex cells, either your sperm or your eggs, depending on whether you're male or female, before you make your sex cells and you only give them one of these chromosomes, your cells can shuffle up the DNA a little bit more. That is called crossover or crossing over, and it's right here. So what happens is a section of dad's chromosome and mom's chromosome, they cross over each other and they switch spots. Okay. This happens during meiosis when you're making your sex cells, your gametes, your sex cells. So why is crossing over a good thing? Well, it makes chromosomes that are they have more variation than the original, so that your offspring now will even have more variation than their parents. And variation is a good thing. It allows organisms to survive. It gives them different types of genes. And these new genes might be better, some cases worse, but might be better and give organisms a better chance of survival. So variation is good. So crossing over, they shift positions, and these are homologous pairs, chromosome 1 from dad, 1 from mom. They flip-flop information. Now, if the information is really close together, whenever a crossover takes place, those same genes always go together. So that is generally why you'll notice people with red hair, pretend red hair is the number 2, and pretend freckles is number 3, and then pretend fair skin is number 4. Whenever that crossover takes place, those genes, man, they all go together. Okay, so 4 and 2 are linked to gene 3 because they're so close together. All right, that's what this information means. So linked genes stay together when a crossover occurs. Farther apart 2 genes are, the more likely a crossover will occur and separate them. So for example, this 4 or this 4 could get separated. These could get separated from the 1 when the crossover happens. So that would allow for variation. Okay, but linked genes, they're the ones that stay together. Mutations, random changes in DNA. In your lifetime, you are going to have a lot of these mutations. Most of them, no, they are not lethal. No, most of them are not gametic or germ cell mutations that happen in your sex cells. You may notice when you go get x-rays, they cover your sex cell area on your body with a very heavy sheet to block out any potential radiation so that you don't have a germ cell mutation and don't have an offspring that has a mutation, which could be potentially maybe lethal. Okay, so there's 2 kinds of mutations. Mutations that happen in your sex cells, germ cell mutations, and those can be passed on to your babies. Okay, there's mutations that happen in your body cells, and somatic cells are body cells. So this is a body cell mutation, and those ones could lead to things like cancer, for example. Okay, somatic cell mutations don't always lead to cancer, no, but they could lead to cancer. Germ cell mutations, why did they put that protective heavy sheet on you when you get x-rayed? So that you don't have a mutation in your germ cells and you don't have issues with babies. Your babies don't have problems down the road. Okay. Some mutations can be beneficial, which means you might have mutated and things got better. You made a protein that works better. That could happen. I would say it's not common, but it could happen. Some mutations don't do anything at all. There's no change physically in the organism. All right. Some could be lethal. Organism could die. Okay. Why are mutations important? They change DNA. And I said earlier, they provide differences or variations. And variations are good because evolution is dependent on differences between organisms. And we'll talk about that later on when we get in our evolution chapter. Now, mutations, I'm going to explain the different kinds. They can involve an entire chromosome, a section of a chromosome, or just one piece, one part, one letter in that code. Okay, so mutations could happen different ways. So let's look at the types of mutations. Now, the first kinds, deletion. Okay, a deletion doesn't mean you're missing a whole chromosome. No, it means you're missing a chunk or a section of a chromosome. So say this is the original chromosome, and now it looks like that. A chunk was X'd out or deleted in that case. Inversion, you have a chunk that looks like this, and then it flip-flopped. Okay, see how this part went there, that part came from there. So there's a flip-flop of information. So, and you'll notice the arrows are showing a flip-flop of information. Okay, inversion, things get inverted. Translocation, well, that means a piece breaks off of one chromosome, and then it reattaches to a whole different chromosome. What term do we use for a different chromosome? Remember, a non-homologous pair. So remember I told you chromosome 1 from mom, 1 from dad, that's a homologous pair. Well, these are chromosomes from different pairs. Notice they're different sizes. So one broke off of one pair from one. So for example, this could be chromosome number 1. This could be chromosome number 18. So a piece of chromosome 1 broke off and attached to chromosome number 18. That's called a translocation. A duplication is when a section gets copied and now you have an extra copy of a section. Okay. Non-disjunction mutations. Now, earlier I mentioned to you about those 3 of that pair number 21. Sometimes when organisms make sex cells, something goes wrong. So when they do this process called meiosis, and you make sex cells that have half the number of chromosomes. So an organism is supposed to have 46 chromosomes. 23 came from mom, a human. 23 came from dad. Well, if mom or dad, let's just say dad gave 22, an extra 21, and mom gave her 23, dad gave, 24, I messed that up. Dad gave 24 chromosomes and mom gave 23. Well, you end up with an organism with 46. I keep messing up. 23 from mom, 24 from dad, 47 is what you end up with. This person is called, someone who has this called karyotype, picture their chromosomes. They have a disease called, why am I drawing a blank right now? They are Down syndrome. Okay. Common. Down syndrome is common. So it's called trisomy 21. They get 3 for pair 21. Do they live? Yeah, they can. Sometimes they don't survive. Generally speaking, if non-disjunction occurs and you make a baby with an extra number 1, they'll die. They will not survive. That will be a baby that was miscarried. An extra number 2, miscarriage. An extra number 3, miscarriage. Number 4, miscarriage. Number 5. I believe you can only have an extra 13 and possibly survive, an extra 18 and possibly survive, an extra 21 and possibly survive, and an extra sex chromosome and possibly survive. If you have any of these extras, an extra 2, death, extra 3, miscarriage, extra 9, miscarriage, extra 10, miscarriage. So non-disjunction, an organism will have basically the sex cells will have an extra chromosome or they'll be missing a chromosome. If you look over here, this individual's chromosomes, they only have one sex chromosome. They only have an X. By definition, they are female. To be male, you have to have a Y. This person would look like a female, and you would not know that if they were sitting next to you, you would not know that they were a female. You would not think anything of them because they're missing that extra chromosome. They would have some health impairments, yes, but you would not know by looking at them that they have a chromosomal abnormality like this. Down syndrome, children with Down syndrome, people with Down syndrome, they have some physical characteristics that you would notice. So you can tell when someone has Down syndrome, they have generally like a rounder face, shorter stature. They're born with a cleft palate. They're born with a lot of ailments. It's called a syndrome because it is a collection of problems. A lot of them don't live a long life. And one of the problems that they incur, because this extra 21, some of them have heart problems too. It's very sad. This person here, this is called Turner syndrome, I believe, or monosomy X syndrome, where you only have one X, basically. They would be female because they don't have a Y. As long as there's an X, no Y, you would have a female here. So non-disjunction, extra chromosome, missing a chromosome. Now, Old Mendel did not know about polygenic inheritance, but we know about it now. What is polygenic inheritance? What it means is that we know that there's certain characteristics that it's not just two factors that cause them. There's more than two factors, maybe three factors or six factors. Many factors can control them. Okay. What I mean by three genes, so that would be 3 times 2, 6 factors are controlling them. So, or 6 genes, and you have pairs of these genes, so that would be 12 factors. What are those things? Skin color is not so simple. It's not like there's many shades of skin color. So there's many genes that control skin color. That's influenced by several genes. Eye color, it's not just one shade of blue, one shade of brown. There's different shades. Height, there's tall, but there's different ranges of tall. Hair color is a mix. Even your hair texture, like how curly it is and how thick it is, there's many genes that control those things. Poly means many gene inheritance. Some things are complex characters. Well, what does that mean? Well, they're influenced by genes, your DNA, yes, and they're influenced by the environment, like how you live your life and what you're exposed to. Let me give you skin color. Not only is skin color polygenic, skin color is also complex. Well, if you have certain DNA, can your skin color that you're born with, can it get darker and can it get lighter? It can. So depending on your exposure to UV light, it can change. UV light would be your environment. Your DNA, your genes would control how dark you can actually get or whether you're going to burn, for example. Human height is also polygenic, but it's also complex. Why? Well, you have certain DNA that says you're going to be tall or short, but if you eat poorly, you're probably going to be shorter. If your diet isn't good and you don't eat enough, you're not going to be as tall. Disease is another factor. Okay. Well, you might have DNA that says you're likely to get cancer. And now you go out, or you're likely to get, for example, any type of cancer. And now you go out and you smoke a pack of cigarettes a day. That pack of cigarettes is your environment. You've induced it upon you. You've brought it around you. It's your personal environment, but it is the environment. That pack of cigarettes combined with your DNA that says you have a history of cancer, not a good situation. Those are called complex characters. So they're environmental and genetic. Now, when we want to look at an organism's chromosomes, we need to extract cells, and usually they want to do it before they're born, so you know how you need to treat the baby when they're going to be eventually born. So amniocentesis is the common one. That's where you poke a needle into the amniotic sac, and there's this fluid floating around. So it basically withdraws fetal cells from the fluid that is flowing around. It can be dangerous. They use a sonogram where they move it around. They want to make sure that they're not poking the baby. There is the potential of poking the baby, but this is called amniocentesis. When did they do it? Usually around 14 to 16 weeks. Then they turn it into a picture of the chromosomes. They want to see, does the baby have an extra 21? Or is the baby missing an X or missing a chromosome? So that's what the karyotype, the picture, will tell you. Chorionic villi sampling, this one's different. This one where they go through the chorion. So they want to take tubes. So they send a suction tube through the vagina, through the cervix, and then they get really close up in this area and they're suctioning out cells they're called CVS or chorionic villi cells. All right. Those contain fetal cells in them, and they can take those and turn them into a picture of the chromosomes called a karyotype, and then they can see. This one is done earlier. Okay, 8 to 10 weeks. This one's done 14 to 16 weeks. New method of treating things, gene therapy. So the whole idea is to insert a new gene into an area where there wasn't a gene or a gene was defected. So you got to get it all the way into the nucleus of a cell. It's not easy to get a gene into the nucleus of a cell and get it to do something or get it to work. It's not that simple to do. Okay, so what have they done? They have used viruses. So scientists have taken viruses and engineered them to not make you sick, because viruses have genes inside of them. They actually do. They have DNA inside them. And when they infect you, they take over your DNA and they screw it up. That's what a virus does. Something like COVID does that, or the flu does that. So they enter the cells, they infect them, they put their genes inside of them, and they screw things up that way. That's what viruses do. Well, scientists have said, hey, wait a minute. Why don't we take out the bad DNA from the virus and insert some good DNA and have it infect people with a virus that is now containing new DNA that is healthy, good DNA. And maybe that DNA can be taken up by the cell and help produce things that the cell needs, and maybe you can use it to fight a disease, or you can use it as disease treatment. This is called gene therapy. So basically, they're using it. The reason it's very temporary, because cells die and they duplicate themselves. When they die and duplicate themselves, this new DNA that you inserted may not be in that new cell that you produce. Okay, so that's why it's only temporary. This is generally done in somatic cells. Why? They're done in body cells. If you did it in sex cells, gametic cells, it's not good. You're trying to change DNA before the baby is born. You're going to be playing a higher power if you did it in that way. I'm not saying it won't happen in the future, but right now it is against the law, and it is considered unethical to do it to germ cells or gametic cells, sex cells. Okay? We're done with today's lecture.